Hi, my name is Brian Searle, and I'd like to tell you about some lessons learned the hard way on acquiring and analyzing data-independent acquisition, or DIA data, with Orbitraps. To begin, this talk is Creative Commons open source license, so feel free to email me for the slides and remix them into your own presentations with attribution. If you have follow-up questions, please either email me directly or tweet at my Twitter handle. I'd like to start out with my acknowledgments first. This work couldn't have been done without Lindsay, Seth, and Mike, and I thank them for all the work that they've put into this. Before I get started, the ISP asked me to show you this speaker disclosure. It just says that I am employed at the ISP, but that I'm also a founder and shareholder of Proteome software. We've heard some about DIA already in this session, but in case you've just come in or need a refresher, DIA is a multiplexing acquisition strategy that repeatedly measures peptides using fixed precursor isolation windows. For example, in this schematic, there are 3 peptide signals with precursors found between 475 and 500 MZ. The peptides in this range are selected by a quadrupole and then co-fragmented together, creating unusually complex ASMS spectra. In this strategy, generally people acquire a precursor spectrum followed by sequential fixed window ASMS. Here, each window is repeatedly measured every 2 to 3 seconds, ensuring that there are enough points across the peak to enable accurate peptide quantification. Let's look at a peptide in this 475 to 500 MZ wide window. At the apex, we might see a spectrum that looks like this. But we have multiple measurements covering this peptide. And we actually acquire, in the end, 3-dimensional fragmentation level data. And we could visualize these fragment ions as chromatograms if you look at them just as the fragment ions themselves. Is this comprehensive quantitation for all peptides? Well, possibly, but there are some complications. First, peptides can't be collision energy optimized based on their charge state. And as I said earlier, multiple peptides are multiplexed together, which highly complicates the spectra. And then finally, quadrupoles don't have perfect transmission across the entire precursor isolation window. So there are edge effects that can suppress signals near the window boundaries. Well, with that out of the way, I want to tell you about how DIA differs on TOF and Orbitrap instruments, and then how to redesign DIA measurements to take advantage of Orbitrap features, as well as to hide their weaknesses. DIA search engines use peptide libraries, and in many circumstances, it is either impossible or impractical to generate these large DDA libraries. I'll discuss how you can rapidly generate DIA-only libraries, and then end with some resources that we built to help acquire high-quality DIA datasets. Modern DIA methods were originally developed and trademarked as swath on time-of-flight instruments. They took advantage of TOF features such as very fast scan rates, and after noticing that peptides tended to clump between 400 and 900 MZ, these 32 fixed window DIA measurements quickly became variable width DIA, where windows were narrower in the sweet spot and commensurately wider at higher or lower MZ. TOF scanned pretty fast, so 32 windows quickly became 40 windows, which became 60 windows, which became 120 windows, with ultimately quite small 5 MZ wide windows for a large range of peptides. Because these windows were so small, many peptides suffered from the edge effects we talked about earlier due to incomplete quadrupole transmission. Considering the actual scans themselves, small margins were added to windows to compensate for the quad isolation effects. Now let's compare that to Orbitrap instruments, which are comparably very slow, collecting MSMS at only 10 to 20 hertz. If we apply that same DIA strategy, well, to achieve a 2.5-second duty cycle, we'd be working with about 25 very wide windows that were over 4 times larger. However, there are some advantages to Orbitraps that this strategy doesn't consider. First, Orbitrap instruments use trapping devices to help use a higher proportion of the ion beam. Measurements are also Fourier transformed, which tends to help filter out a bunch of this low signal noise. Additionally, since the release of the QE Plus in 2013, they've incorporated a segmented quad, which significantly improves transmission efficiency across the entire window. Well, let's try to build a method from the ground up that uses these features to our advantage. First, we're collecting too many scans. Rather than going up to 1250 MZ, we should take advantage of that plot from earlier that shows that most peptides fall within a narrower range, such as this 400 to 1000 MZ range that I'm showing here. There will be some peptides that live above that range, but I suspect that many of those will be found at higher charge states within the 400 to 1000 MZ wide range. Well, looking at HeLa data sets, we ran replicate injections with 5 different windowing schemes with increasing window widths from 500 to 900 MZ, all the way up to 400 to 1200 MZ. While widening this range helped to a point, as windows got much wider, the detection rates dropped, no matter what library search strategy was used. We found that this 400 to 1000 MZ range was the optimal sweet spot. However, we only really know that this works for normal tryptic peptides. Other types of peptides, so for example these phosphopeptides, will likely have different optimal ranges. So considering this phosphopeptide library, that would indicate that maybe searching or considering between 500 and 1100 MZ would be a better acquisition strategy. While we can't actually collect spectra faster, we can use undersampling math to make it seem as if we were. So considering the normal window boundaries here, we can stagger every other cycle by 50% using a technique called compressed sensing. Let me give you a feeling for how that works. Let's consider this 600 to 624 MZ fragmentation spectrum from a 24 MZ wide window experiment, and then consider the previous and next scans from the previous and next cycles. Some ions, such as this red ion, are present in all of the scans, and as a result, we can't really say much about that one. However, some ions, here, for example, in green, are only present in the 588 to 612 MZ previous and next spectra. And these can only come from peptides with precursors between 600 and 612. And we can use linear algebra to create a new spectrum just for these ions. Similarly, there will be other fragment ions that are present only in the 612 to 636 MZ previous and next spectra. And we know that these can only come from peptides with 612 to 624 MZ precursors. And now instead of having 24 MZ wide windows, after demultiplexing, we now have 12 MZ wide windows, absolutely 100% free, with a little extra computational processing. However, this type of math is severely affected by noise. That said, as we discussed earlier, Orbitraps produce relatively little background noise, and this approach works remarkably well. Now, here there's a little bit of a problem. This staggering approach needs to have fixed window boundaries throughout the experiment and is significantly complicated by the addition of margins that hang over that edge. Here we can lean on another property of Orbitraps to help with this. Peptides are all made of the same stuff: carbon, hydrogen, nitrogen, oxygen, sulfur. There are only so many ways to configure amino acids, and as a result there are sub-MZ regions of the precursor space where you simply cannot have a peptide with that MZ. Here I'm showing that with the distribution of +2 and +3 precursors from tryptic peptides in the human SwissProt database. These peptides cannot fall in these forbidden zones, which are about 1/4 MZ wide. And coincidentally, this also happens to be about twice the edge fall-off of the segmented quadrupoles that are in most current Orbitraps. By placing window boundaries at these fractional masses instead of the traditional nominal masses, we can completely remove the need for window margins. These boundaries actually hold up pretty well when compared versus the pan-human library here with the SwissProt library. In general, they match up exactly as you might imagine. But it should be noted that non-tryptic peptides and peptides with PTMs may have somewhat different characteristics. Phosphopeptides, for example, have optimal boundaries that are shifted over by 0.18 MZ due to the addition of the extra phosphate. So let's use the remaining time we have here to talk about building DIA-only libraries. DDA libraries have been intrinsic to the success of DIA and are used to determine the properties of peptides that you want to measure with your instrumentation. However, they are imperfect in several ways. First, these libraries require a significant amount of sample for offline fractionation, as well as time and effort. And that can be impractical for some experiments and impossible for others. Second, DDA fragmentation on Orbitraps is charge state optimized, while DIA fragmentation inherently cannot be. This results in small fragmentation variations that can have real-world effects. Third, because offline fractionation changes the peptide background and the matrix that each peptide sits in, it can also subtly change retention times too. Rather than generate DDA libraries for our DIA experiments, we have opted to use an alternate method to generate DIA-only libraries. This method relies on gas phase fractionation, which was originally developed and applied to DIA in the Goodlet lab over 10 years ago. The principle is to break up the acquisition of single samples into multiple runs that cover the same total MZ space with smaller windows. This type of fractionation is interesting because it doesn't involve any extra offline work, and it only requires setting up 2 DIA methods with different windowing schemes. We have extended this out further to 6 gas phase fractions that each cover 100 MZ wide. These runs have 4 MZ wide windows, which after staggered demultiplexing results in effectively 2 MZ wide windows. In a sense, these datasets have the same precursor isolation as DDA datasets and the same high quality measurements as targeted PRM experiments. except that instead of targeting specific peptides, they actually target every peptide from 400 to 1,000 MZ. This strategy would be impractical for almost every type of experiment if you had to do every sample in this framework. However, it uses 6 micrograms of material to generate, if we use it on a pool, to generate a library based on that pool, which can be used for future DIA experiments to do analysis. These 6 micrograms of material and these 6 injections is dramatically less material and effort than that required for traditional offline fractionation with DDA methods. We collect these 6 injections on a sample pool made from a sub-aliquot of each of our quantitative samples. We then search them using either a FASTA database directly with the Pecan search tool or with predicted fragmentation patterns using the PROSIT tool for every possible tryptic peptide in that database. With effectively 2 MZ wide windows, even if the retention times and fragmentation patterns are a little off, these injections are so clean that it's very easy to detect peptides from them. And now that we know the specific DIA-based fragmentation and the exact dilution characteristics for each peptide on our instrumentation in our matrix, it's much easier to look up these peptides in wide window DIA experiments that we do afterwards, where the chromatographic information from the gas phase fractionation and the wide window DIA is exactly the same because it's working within the same matrix. And the fragmentation patterns are exactly the same because they're configured the same way because they're both DIA in each case. I should note that the 6 gas phase fractionated injections don't share many peptides, so it's important that there's as little retention time deviation between them as possible. We recommend waiting for the column to break in and collect the gas phase DIA runs after at least 6 normal injections within the same matrix. While it's difficult to retention time align between the gas phase fractionated runs to build the library, that said, after it's built, it's actually pretty easy to align the library to those early normal injection runs and correct any retention time deviations that happen to be in those files. But with these libraries, which again only require 6 injections and 6 micrograms of a sample pool, we can achieve excellent results. Compared to sample-specific DDA libraries using offline fractionation in yellow, we can use the 6 gas phase fractionated injections to essentially empirically correct this library with more exact retention times and fragmentation patterns in orange, and achieve higher detection rates in both yeast on the left and human cells on the right. Similarly, while PROSIT predicted libraries in light blue underperform when compared to fractionated DDA libraries, the performance is quite high after correction with the 6 gas phase fractionated injections. We've developed a bunch of resources to help people start collecting data on Orbitraps using our framework, and I wanted to share them with you. First, much of what we've discussed here has just been published in a brand new preprint at MCP. We've been working on to build a quick start guide as well as a spreadsheet of recommended DIA settings for every QE or tribrid instrument to date. And finally, both the slides that are shown here as well as the software to implement these methods are freely available at this bottom URL for the Encyclopedia Software Tools Suite. Thanks again for your attention, and I look forward to your questions.